Welcome back to another video in which we will continue our discussion of cytokine signaling. And in this video, we're going to become acquainted with yet another family of cytokines, this family known as chemokines, a subtype of cytokines that are particularly important for the induction of cellular movement. So you may have heard of the term chemotaxis before. You can sort of derive from that name the idea that we have some kind of chemical messenger, chemo, inducing movement. Taxi, you can sort of imagine what a taxi does. It drives around. So chemotaxis is the sort of chemical induced movement of cells. And you can even get from the name of chemokines themselves that they are chemical messengers, and the kine root here sort of gives us the idea of something that's kinetic, it's moving, it's in action. And the induction of chemotaxis is an especially important feature of the immune system. Chemokines play a very important role because immune cells need to get around the body and they need to be recruited, especially to sites of infection and sites of injury. And so they do this by establishing concentration gradients as they diffuse away from their cellular source. So in this simple example that we have here, we can see an infected cell. There's viruses in it. And infected cells, for example, produce chemokines typically in large quantities. And as this cell secretes the chemokine, the concentration of that chemokine is going to be greatest at the source, nearest to the cell. And as the chemokine diffuses away, the concentration is going to diminish. And cells that detect those chemokines, that have the cognate receptors for those chemokines and can sense them, are going to be attracted to move up that concentration gradient, and that's going to bring them into proximity to the source, in this case the infected cell, so that they can mount whatever effector function is needed in that scenario. So this is just a general overview of how chemokines induce chemotaxis and what the purpose of that is. Let's think now a little bit more deeply about chemokines. Like many of the other chemokine families that we've been introduced to so far, chemokines also have subfamilies. And the subfamilies of chemokines are notoriously a little bit difficult to keep track of. But I'll give you a little bit of a cheat sheet here, which is to say that there are 4 subfamilies of chemokines. They are the CC family, CXC, CX3C, and the XC family. And these names do not derive from any functional difference within these families, but in fact from a structural feature of their primary amino acid sequence. So in the N-terminus of these chemokines, there are conserved cysteine residues. These confer structural integrity to the chemokine because they form disulfide bonds with each other. But if you look at those cysteine residues, they have sort of different patterns depending on the type of chemokine that we're dealing with. So chemokines in the CC family have 2 cysteines that are right next to each other. They are directly physically opposed. And so we get the CC name from that. However, some chemokines have a cysteine and then an intervening random amino acid, which is designated by the letter X, followed by another cysteine. And so we call chemokines of this family CXC chemokines. Using a similar logic, chemokines that have 3 intervening amino acids between their 2 N-terminal cysteines we refer to as CX3C. And then finally, a small number of chemokines that only have a single cysteine in their N-terminus we refer to as an XC chemokine. And sometimes this is even just referred to simply as the C family. But ultimately, these 4 families are going to sort of lend, I should say that this nomenclature of CC and CXC and so on, this is how we end up naming chemokine ligands and receptors. So you can notice that there's a further convention where chemokine ligands of the CC family, we simply add L to the CC designation. So chemokine ligands are always CCL. Whereas receptors for CCL chemokines are going to have an R, a CCR. So CCLs bind to CCRs, CXCLs bind to CXCRs, and so on and so forth. And this is important because chemokine ligands will only bind to a receptor in the same family. You don't get any cross-reaction, for example, between a CCL chemokine and a CXCR receptor. They'll always sort of keep it in the family in terms of the specificity of their interactions. So these are the 4 families, and we can make some additional notes about the way chemokines behave, particularly their binding interactions. And a major feature that's worth knowing about chemokines is that they exhibit what we call promiscuous receptor binding, meaning that there is typically a low degree of specificity for any given chemokine receptor and the ligands that it can bind to. Many chemokine receptors have multiple ligands that they interact with, and we refer to this idea as molecular promiscuity. So you can see an example of that here. We have the chemokine receptor, CXCR1. Again, notice the R in the name indicating that it's a receptor, and it's of the CXC family. And its N-terminus, the chemokines of this family have a cysteine with an intervening amino acid and then another cysteine. The two ligands that bind specifically to CXCR1 are CXCL6 and CXCL8. Notice again the L's here because these are the ligands. And notice that the families match. These are CXC chemokines binding to CXC receptors. Something else you can notice here is that the numbers, however, do not match. The numbers really have nothing to do with each other. Many of these chemokines, the numbers are just simply the order in which they were discovered. And so there's no sort of indication about how an individual chemokine might interact with the receptor from the number alone. So you can see that for CXCR1, that it can bind to both CXCL6 and CXCL8. CXCR2 is even more promiscuous. It has at least seven well-established chemokine ligands, 1, 2, 3, 5, 6, 7, and 8. CXCR3 similarly has 3 established ligands. The unique member here is actually CXCR4, which is specific for CXCL12. So this is a rare example of a non-promiscuous interaction. There's actually a high degree of specificity of CXCR4 for CXCL12 comparatively to the other chemokine receptors in this family. Now, there are other CXCRs that are not shown here, and then of course there are other receptors and ligand pairs in all of the other chemokine families. It's not all that helpful in an introductory course just to kind of memorize by brute force which ligands go with which receptors. This slide is mostly meant to make a conceptual point about how the nomenclature allows us to determine kind of what is going to bind to what, and to demonstrate that there is a high degree of overlap and redundancy in chemokine signaling, and that comes about because of this promiscuous receptor binding. So many of these chemokines can accomplish the same end result because they activate a shared receptor. However, some of these chemokines may, for example, exhibit higher affinity for that receptor than others. Some of these chemokines are going to be different in terms of which cell types might express them and in what parts of the body and maybe at what point in time following an infection. So there are reasons we have so many chemokines, but ultimately they're going to sort of transduce very similar signals using the same receptor. You may also notice that the shape designated by these cartoons corresponding to these chemokine receptors are evocative of a G protein coupled receptor. They sort of have these 7 transmembrane passes. Even if you don't recognize it, I'll go ahead and tell you that most traditional or typical chemokine receptors are in fact G protein coupled receptors. And they have this in common, if you'll remember, with anaphylatoxin receptors. They are receptors that also use G proteins intracellularly to signal. Now, once again, I'll say that the purpose of our class is not to really dive into the specifics of G protein signaling, but I'll remind you that G proteins transmit a lot of very complex intracellular signals using a variety of second messenger molecules, and they exert highly pleiotropic functions on the cell in which they are signaling. So some of those pleiotropic functions that are downstream of chemokine receptors are, of course, cellular movement, which is primarily orchestrated through cytoskeletal reorganization. So through the activity of molecules like Rho GTPases, G proteins can induce the cytoskeleton to reorganize, and that's critical for a cell to be able to move through its environment. Ultimately, movement is all about changing the structure and the shape of the cell as it sort of maybe it extends a foot process and drags itself along, or maybe it squeezes itself flat so that it can perform diapedesis through the walls of a blood vessel. All of that requires cytoskeletal remodeling. However, even though that cellular movement is the most sort of unique and the effector function that we primarily associate with chemokines. They do have other functions. Chemokines are especially important in many contexts for inducing cellular survival. They do that through a molecule known as AKT, and I've simplified this here simply to say that AKT suppresses death. Particularly, it suppresses apoptosis. And so the suppression of apoptosis through chemokine receptors is especially important during development. And that's actually kind of a non-immune function of chemokines. But also a critically important thing that they accomplish. Finally, the G protein signaling in traditional chemokine receptors also very potently alters the gene expression of the cell, and this may also be important for mounting various effector functions in the context of infection. So chemokine receptors are G protein coupled, and they exert pleiotropic functions. There are some other sort of specialized features of chemokine signaling, though, that make this family stand out as somewhat unique. One is that more recently we've discovered that in addition to that complement of traditional chemokine receptors that exist that are G protein coupled, there are also chemokine receptors that we refer to as atypical. And these atypical receptors typically, I should not use the word typically. These atypical receptors, they often look like G protein coupled receptors structurally. Notice that this purple atypical receptor here has the same structure with those 7 transmembrane domains, but it is not G protein coupled, which is what makes it atypical. So despite being sort of physically similar, atypical receptors either do not really transmit intracellular signals, or if they do so through G protein independent mechanisms. And they do a variety of other things that tend to regulate chemokine signaling rather than promoting the traditional effector responses of chemotaxis and so on. I'm giving you an example of a particular atypical chemokine receptor on this slide. This is just one example to sort of illustrate the point. We saw before that CXCL12 is a chemokine ligand which binds to the typical or traditional chemokine receptor known as CXCR4. The atypical receptor for CXCL12 is known as CXCR7, which actually more recently has sort of been renamed ACKR3 for atypical chemokine receptor 3. But these names are functionally interchangeable. They represent the same molecule. For simplicity, I will probably just say CXCR7. When CXCR7 binds to CXCL12, that is when this atypical receptor binds to the chemokine ligand, rather than transducing a signal through G proteins, instead that induces endocytosis of the receptor ligand complex. It's internalized into an endosome, and that endosome ultimately fuses with lysosomes. Within the lysosome, the CXCR7 receptor lets go of that CXCL12 ligand molecule, and it's now exposed to the acidic environment of the lysosome, as well as the various proteases that we've seen are enriched in lysosomes, and it's degraded ultimately, and we get rid of that molecule of CXCL12. Well, following that, the CXCR7 can then sort of leave the lysosome back through the endocytic pathway and ultimately be trafficked back to the cell surface, sort of go back to where it started from. But notice now it no longer has CXCL12 attached. The purpose of this is something that we refer to as scavenging. CXCR7 can operate as a scavenger receptor, and by plucking CXCL12 out of the extracellular environment, dumping it into the lysosome, and then sort of coming back for more, this is a negative regulation of CXCL12 signaling, because the more that it does that, the less CXCL12 is left in the extracellular environment, and therefore the less CXCL12 there is to activate the typical receptor to induce G protein signaling. So this ultimately is a process that we might refer to as neutralization of CXCL12 in terms of its abundance, at least, in the extracellular environment. So if we want to turn down the heat a little bit on CXCL12 signaling, we can use an atypical receptor, a scavenger receptor, to decrease its abundance. And so, again, this is just one example using CXCL12, but there are other atypical chemokine receptors, several of which have this scavenger function. And I should say that atypical receptors are much more recently described in the research literature, and so we're still sort of understanding some of the additional regulatory functions that atypical receptors have in terms of chemokine signaling. So to finish this video, I want to talk about one particular scenario and maybe even one particular anatomical location where chemokines, and especially the typical activity of chemokines involved with cellular movement and chemotaxis, where it's really important. In order to sort of make this a little bit less abstract, we've learned that chemokines induce cellular movement, but why does that matter? How do we use that in the process of an immune response? Well, there are many examples that I might give you, but I'm going to give you one particular example that I think is especially important and underscores the way that chemokines sort of orchestrate various key features of any given immune response. For this, let's take a trip back into the lymph node. We haven't been here in a while. And within the lymph node, which is a secondary lymphoid organ, you may recall that there were anatomical locations like the primary follicle, and the primary follicles were enriched with naive B cells, here shown in blue. In contrast, we had paracortical areas, or the paracortex, which was enriched with naive T cells. And these are the locations where these naive lymphocytes sort of hang out until they see their antigen and become activated. But one thing that we haven't discussed yet is how is it that these naive cells come to reside in these locations? How do they get there? And equally importantly, how do they stay there? How do they stay segregated in these discrete anatomical sites? Why don't naive B cells cross over into the paracortex and vice versa for naive T cells? The answer to that is chemokine signaling. So within these two locations, there are unique chemokine signals that exert strong chemotactic pressure for these two populations of lymphocytes to come in and to stay. So let's talk about a couple of major examples. Within the primary follicle, there are cells known as follicular dendritic cells. These are specialized dendritic cells that live in primary follicles, hence they are follicular dendritic cells. And they produce a particular chemokine known as CXCL13. CXCL13 is specific for a particular chemokine receptor known as CXCR5. And the cells that express CXCR5 happen to be naive B cells. So as a naive B cell is passing through a high endothelial venule, passing through the lymph node, it has a receptor that allows it to detect CXCL13 in its environment because it can bind to it. And as it starts to encounter CXCL13 that's emanating from these follicular dendritic cells, that's going to be a strong chemotactic cue for it to move towards the source of that chemokine. Ultimately, that's going to bring it into the primary follicle. And not only is it going to bring it into the primary follicle, it's going to make it want to stay. As long as it has CXCR5, it's going to have a strong attraction to any location where there's a lot of CXCL13, which describes the primary follicle. And so it's not only a matter of migration into the tissue, but also the establishment of residence within that tissue. Similarly, naive T cells respond to chemokines known as CCL19 and CCL21. The cognate receptor for which is CCR7. So naive T cells express CCR7, and within the paracortex, there are reticular fibroblasts, which make a lot of CCL19 and 21. So a similar concept plays out here. The naive T cells, as they're passing through the high endothelial venule, they move towards the area with the highest concentrations of CCL19 and 21, and that allows them to establish residence within the paracortex. Now, the reason that these cells don't cross over is that naive B cells express CXCR5, but they do not express high levels of CCR7. So they don't have that signal pulling them into the paracortex. If they had both, they would be a little bit confused if you don't mind anthropomorphizing them a little bit. They would have sort of two signals pulling them in opposite directions, but because they exclusively express one receptor and not the other, they sort of know where to be and they know where to stay. Similarly, naive T cells express CCR7 but do not express high levels of CXCR5. An additional thing that happens here, which is useful to understand, is that T cells and B cells don't want to hang out in the lymph node forever. Eventually, they might see their antigen and no longer be naive, and they become activated. And when they become activated, they need to leave the T cell and they need to migrate out into other tissues in order to exert their effector functions. So when a T cell or B cell sees its antigen and becomes activated, a major thing that happens downstream of the activation of their antigen receptor is going to be the downregulation of these particular chemokine receptors. So when a naive B cell becomes activated and sees its antigen, it's going to undergo a germinal center reaction to proliferate, but ultimately it's going to lose expression of CXCR5. Therefore, it's going to become insensitive to all this CXCL13 in the follicle environment. It's going to sort of be able to then to escape the gravity of CXCL13, leave the lymph node through the efferent lymphatics, and travel out into the body. Similarly, when naive T cells become activated, they downregulate their CCR7, which allows them to escape this chemotactic hold and migrate out into the body to accomplish effector functions as well. Okay, with that example being finished, let's summarize what we've learned in this lecture about chemokines. We saw that chemokines direct the migration of immune cells by establishing chemotactic gradients, and this is one of the primary effector functions that we associate with this family of cytokines. We saw that there were 4 major families of chemokines, the C, CC, CXC, and CX3C, and these are determined by the spacing of cysteine residues in the N-terminus of these chemokine ligands. And within these family, we have a special nomenclature in which we designate the ligands with the letter L, which follows whichever family we're in. So in the CXC family, we attach L for the ligands and then usually a number, whereas the receptors are designated by the letter R. So in this example, CXCR3 would be a receptor, CXCL10 would be a ligand. Finally, chemokine receptors have pleiotropic functions, and we saw some examples of those. They included the suppression of cell death, cytoskeletal remodeling, which is related to cellular movement, as well as the induction of gene expression. And this is accomplished through the activity of G protein-coupled receptors. And we said that most traditional or typical chemokine receptors are G protein-coupled. However, there are less well understood atypical chemokine receptors as well, many of which have scavenger function. And in general, these atypical receptors are not G protein coupled and instead exert regulatory action on chemokine signaling. So that does it for chemokines. We'll have one more lecture in this module where we will talk about pattern recognition receptors. See you there.